this lesson, we're going to talk about membrane potentials and the role that they play in nerve transmission. So first, what is a membrane potential? Well, anytime you hear the word potential related to nerves, I want you to think electrical charge. So the membrane potential is really just the electrical charge across a membrane. Now in the nerves, we have a resting potential and we have an action potential. This should be pretty self-explanatory. The resting potential is when nothing is happening. The nerve is just resting. Now we'll look at this more in a minute, but know that the resting membrane potential or the resting electrical charge in the nerves is negative 70 millivolts. So this just means that it's more negative on the inside than on the outside. The other type we have is an action potential. So as you can see, this is where an action is happening. So an action potential is sending a stimulus in order to create a response somewhere in the body. Now the charge in an action potential is more positive. And we're going to look at specifics in just a minute. So when I say the resting potential is about negative 70 millivolts, what I mean is that it's more negative on the inside than it is on the outside. But since everything equals out based on concentrations, not charges, it's actually at equilibrium that way. So having that negative 70 is actually where we want to stay. So let's look at some of the factors that contribute to this negative membrane potential. First is the sodium-potassium pump. This pump uses ATP or energy to force sodium out of the neuron and potassium into the neuron, and that's usually against the concentration gradient. So for every 3 sodiums that we kick out of the cell, we're going to bring 2 potassiums in. Now, both of these ions are positively charged. So what you're seeing is that more positive ions are leaving the cell than the ones that are coming in. So that makes it more positive on the outside, therefore more negative on the inside. Now, we also have ungated leakage channels. And leakage is the term that you're going to see most often. So you have leakage channels for both sodium and potassium in the membrane of the neuron. And so what we see is the potassium diffusing out of the cell actually diffuses out about 100 times faster than the sodium is entering the cell. So again, we have more positive on the outside than we do on the inside. So that is what gives us this net negative charge on the inside of about negative 70 millivolts or so. Now these ions are constantly moving back and forth so they can kind of keep it right there at equilibrium. So again, this is our resting potential. This is right where we want it. The nerve is just sitting there and waiting for something to happen. Now, when it's time for an action potential, we're actually going to see these gated channels come into play. And gated just means that they have to have certain conditions to be true in order to open. So let's look at what happens there. So an action potential starts with just some local depolarizations or local potentials, or some people will call them generator potentials. So local depolarization. So if this is our big axon, you're really just talking about right here, right in that one little spot. So you have some kind of stimulus, like a chemical signal, heat, cold, etc. And that causes some of these sodium gated channels in this one little spot to open. And when that happens, we now have more sodium coming into the cell than we had before. And so instead of it being super, super positive out here, it actually is relatively more positive on the inside. It's only moderately more on the outside. Now, if nothing else happens, these gated channels close and the cell will just kick out some more potassium to kind of repolarize everything back to that resting potential. However, if we have more stimuli or a stronger stimuli, these gated channels stay open and that sodium just starts to flood into our neuron. And that allows our membrane potential to get more and more positive on the inside until we hit what's called the threshold voltage. Now, the threshold voltage is what starts that action potential and starts it moving down the axon. So instead of just this local depolarization, you actually get a full action potential that moves itself all the way down the length of the axon. So let's look at what happens in an action potential once that threshold stimulus is reached. and look at that sequence of events. So once we reach that threshold, it causes those sodium-gated channels to fly open and sodium rushes into the neuron. So that means the inside gets more and more positive. This process is called depolarization. Once the membrane is fully depolarized, those sodium channels will close and the potassium-gated channels will open. So that actually allows potassium to rush out of the cell. So the membrane now, instead of being super positive, starts to get less and less positive and starts to return back down towards that resting potential. That process is called repolarization. We're taking it back to where it was. But what happens is that before those potassium channels close, more potassium leaves the cell than sodium is coming in. And so that membrane potential actually goes more negative and past the resting potential. This process is called hyperpolarization. Hyper means more, so it goes beyond where it should have. So it's more negative than it was. So once all those gated channels close, the sodium-potassium pump will help to restore everything back to normal, and concentrations will return to that equilibrium and that resting potential. So let's visualize this kind of all together. So here's our resting potential at negative 70 millivolts. We have to get enough stimulus to bring that membrane potential up to threshold. So if we only get these tiny little stimuli and we never get there, we only get these local depolarizations. Okay, this is what I mean up here when I say the all or none law. You're either just going to get tiny local depolarizations or you're going to get a full send action potential, okay? So if we get enough stimulus and the membrane potential gets to threshold, that's when we see the action potential start. We see depolarization as the sodium rushes into the cell, makes everything more positive. Then we see the sodium-gated channels close, potassium-gated channels open so that potassium can rush out. That is our repolarization process. So it brings it back down towards the resting potential. But because we see more potassium out than we saw sodium in, we're going to see this hyperpolarization process where it becomes even more negative than it was. Now you'll notice here during this hyperpolarization phase, there's what's called a refractory period. That means that no other stimulus would ever be strong enough to get us all the way up to this threshold line. So it's a refractory period because it cannot send a new action potential during that time. Then we have the sodium-potassium pump kicks in and equalizes everything back out to where we want it to be, brings us back to our resting potential. So resting, get enough stimulus to threshold, depolarization, repolarization, hyperpolarization, and a return to resting state. Now, this is just a picture of what's happening at one point at a time on the axon. But in order to send a signal, we actually have to see this process continue and repeat itself down the length of the axon. That process is called propagation or conduction. So the action potential moves like a wave down the length of the axon. Now you can see this neuron on the left. It kind of just sends like a wave, kind of like the wave at a baseball game. Now the difference between these two axons is whether or not a myelin sheath is present. If you remember from the neuroanatomy lesson, the Schwann cells will wrap around the length of the axon and create this myelin sheath. And then you have these nodes of Ranvier between each cell. These allow the action potential to move faster and smoother down the length of the axon. You can see the difference in speed and smoothness side to side here. So when this conduction happens like this with the myelin sheath, where it jumps from node to node, it's actually called saltatory conduction, which just means jumping. So it's jumping from node to node, goes faster and smoother. So let's recap what happens with membrane potentials. Remember, that just means the electrical charge across the membrane. So the resting potential in a nerve cell is when nothing is happening, and it's usually about negative 70 millivolts. The things that control these charges are basically the movement of sodium and potassium across the membrane, both with the sodium-potassium pump as well as with gated and ungated channels. Now, once it reaches the threshold, it will start an action potential, which is when a signal is actually being sent down the length of the axon. That process includes depolarization, repolarization, a brief period of hyperpolarization, which is when we see that refractory period, and then a return to resting potential. And that whole process propagates or conducts down the length of the axon like a wave so that we can send the signal where it needs to go. All right, guys, that's it for the basics of membrane potentials in the nervous system. Make sure that you check out all the resources attached to this lesson. Now go out and be your best selves today, guys. And as always, happy nursing.